Okay, continuing on with chapter 10, here we're going to cover section 10.4, cell differentiation. From one cell to many. So how do cells become specialized for different functions? So how do you have eye cells and muscle cells and cardiac muscle cells? So during the development of an organism, cells differentiate into many types of cells. All organisms start out in life just as one cell after the sperm fertilizes the egg or the oocyte. Most multicellular organisms pass through an early stage of development called an embryo, which gradually develops into an adult organism. During development, an organism's cells become more differentiated and specialized for a particular function. So, for example, a plant has specialized cells in its roots, so cells that are characterized by roles that roots have to play, cells that do the roles that a stem has to play, and same idea with leaves. So the process by which cells actually become specialized is known as differentiation, so to become different. During development, cells differentiate into many different types and become specialized to perform certain tasks. So differentiated cells carry out the jobs that multicellular organisms need to stay alive. In some organisms, a cell's role is determined at a specific point in development. So you can actually map out the differentiation in an organism. So in the worm C. elegans, the daughter cells from each cell division follows a path or specific path toward a role as a particular kind of cell. So here we have cells that are for the nervous system. By the fifth cell division, cells in the nervous system begin to differentiate. Here in the cuticle, by the 8th cell division, some of the cells that secrete the worm's outer covering begins to differentiate. And over here for the pharynx, after 9 to 11 cell divisions, the cells in the feeding organ differentiate for the pharynx. When you're looking at differentiation in mammals, cell differentiation in mammals is controlled by a number of interacting factors in the embryo. Adult cells generally reach a point at which their differentiation is complete and they can no longer become other types of cells. So specifically looking at stem cells here, stem cells are unspecialized cells, so they haven't been given their instructions yet of what to do, from which differentiated cells develop. These are known as stem cells. One of the most important questions in bio is how all these specialized differentiated cell types in the body are formed from just a single cell. Biologists say that such a cell is totipotent, literally able to do everything. So toti kind of sounds like totally, in form of all the tissues of the body, or to form all the tissues in the body. Only the fertilized egg and the cells produced by the first few cell divisions of embryonic development are truly totipotent. So when you look at human development, after about 4 days of development, so after 4 days of fertilization, a human embryo forms into something called a blastocyst, this is what a blastocyst is. A hollow ball of cells with a cluster of cells inside known as the inner cell mass. The cells in the inner mass are said to be pluripotent, which means that they are capable of developing into many, but not all, of the cell body types. So here we have the inner cell mass, where you can see that they can differentiate into the pancreatic cells, heart muscle, blood, nerve cells, bone marrow, for example. So this is kind of a similar image that we just saw on the last page, but stem cells are unspecialized cells from which differentiated cells develop. There are 2 types of stem cells. There's embryonic and adult stem cells. So embryonic stem cells are found in the inner cell's mass of an early embryo. Embryonic stem cells, again, are pluripotent. Researchers have grown stem cells isolated from human embryos in culture. Their experiments confirmed that was where this idea came from, that embryonic stem cells have the capacity to produce most cell types in the human body. Adult stem cells are found in mature, not embryos, but mature organisms. So adult organisms contain some types of stem cells. Adult stem cells are multipotent. They can produce many types of differentiated cells. Adult stem cells of a given organism or of a given organ or tissue typically produce only the types of cells that are unique to that tissue. So that's like multipotent means the idea that like an organ, the stem cells found in that organ can form only the types of cells that are in that organ. So they can, they're more specialized in that way or limited. So stem cell research. So possible benefits and issues. There's lots of issues associated with stem cell research. Stem cells offer the potential benefit of using undifferentiated cells to repair or replace badly damaged cells and tissues. Human embryonic stem cell research is controversial because the arguments for it and against it both involve ethical issues of life and death. So that's where we come up with issues in science. Whenever you have ethical reasonings, you have to keep those in mind for sure. So potential benefits, stem cell research may lead to new ways to repair the cellular damage that results from, like, for example, a heart attack, a stroke, spinal cord injuries. So one example is the approach to reversing a heart attack damage. So here you can get stem cells from that are found in the bone marrow and remove those from a patient's hip. So it's like a hip bone. They took the bone marrow out. They inject the stem cells into the damaged area of the heart and the environment of the heart, so the surrounding cells kind of stimulate those stem cells to turn to differentiate into new heart cell muscle cells, in essence fixing the injured area here of the heart. Obviously, ethical issues are involved here, so most techniques for harvesting or gathering embryonic stem cells would cause destruction of the embryo. Either a lot of times they're either coming from voluntarily aborted fetuses or embryos or spontaneously aborted embryos, which would be like a miscarriage. Government funding of embryonic stem cell research is an important political issue. So there's, groups seeking protection for embryos would oppose this type of research because it's considered unethical. And then other groups that support the research are supporting it as an essential to saving current human lives. And so they would view that, opposing it, as unethical, that's restricting the research, that's preventing human lives, humans that are living from getting the proper help that they need. And the groups that oppose it are against it for hindering embryos that had the potential for life from not having that life. So there's lots of ethical issues that have to be worked through for this case. So this is among the all sorts of other ethical issues with all sorts of other things in science. So like from the first chapter, we talked about ethics in science. So it's a very interesting topic. Okay, that is it for chapter 10.